Today, I spoke with Dr. David Bowran. David's an expert in herbicides and he's a breeder as well, just quietly. And so we touch on that. He's an enthusiast for climate change happening and how we need to adapt for that. I'm not as concerned as he is, but you'll see we clash a little bit, but delightfully. David always speaks his mind. I really respect him for that. And he was a great support in the no-till revolution, which of course I'm no-till Bill. So he's been very helpful in agriculture in many aspects, and I think you'll enjoy his very intense mind. He loves to read scientific papers and pull gems out. We touch on DDT as well, so there's quite a bit in here that I believe you'll enjoy. Thank you for watching. Well, welcome, Dave. Good to have you here with your long experience history in agriculture, particularly in herbicides.
Bill?
Yeah, and what got you into agriculture?
What got me into agriculture? Well, I come off a family farm, so that was a great start.
Where was that?
So my parents originally were at Carnama, wheat and sheep, but then we went to Bindoon and ended up with sheep and oranges, and then into grapes. So I actually had quite a lot of time doing different horticulture stuff as well as sheep. And then as a kid, I used to be fascinated by all the different crop types. And I used to pick the barley grains out of the oats and grow barley just to see what it looked like, to see why it was different. Parents thought I was a bit mad, I think. They used to grow it in the old chook pen and see how it would grow, and tried all sorts of interesting little experiments. And I was only about 13 when I was doing that.
Okay.
So I already had a bit of a bent. And then I read a science encyclopedia that Mum got us, and that talked a bit about agriculture in there. And so, yeah, I already had, strong leaning towards agriculture.
You would have got pretty good marks at school, I'd imagine.
Certainly at high school, I was in the top 3 in my class up until year 10. When I went to senior high school, I dropped back a bit because there were a few really brainy kids I had to compete with. But I loved my chemistry. It was what really, turned me on was chemistry, because we didn't have biology, being in science stream. The biology was for the lower science stream, whereas the top science stream was chemistry and physics. So I used to absolutely adore chemistry.
And same reason as me, because you could look around the room and say, I know which molecules are in each of these different things. Is that one of them?
Yeah, not really. I was more interested in the actual reactions themselves and understanding why things were going on. Simple things like, one day we walked into class and we could hear this pop, 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 pop. And we couldn't work out what it was. And my science teacher walked in and said the other science teacher was making nitrogen triiodide, which of course spontaneously erupts. So it's actually used to make a very cheap explosive. And he'd made some, and the trouble is that it exploded and it had gone off all into the room. So the ceiling was covered in all this
yellow, orange
stain, and it was going off. As it dries out, it spontaneously goes off.
Wow. They were the good old days.
They were the good old days when you could do those things. And of course,
occupational health and safety would say, don't go near it.
Nowadays, the chemistry teachers get a real rap over the knuckles if they do anything like that. Anyway, I love chemistry.
So then you went to uni straight away and did agriculture?
After I finished, I went to uni. I had a few choices. What am I going to do? I don't know. Like chemistry, I'll put agriculture first. At least I have chemistry as a backstop. But of course, they wanted me to do agriculture. I got my marks were high enough, so I just went in and did agriculture.
And then once you finished your Bachelor of Ag at UWA?
Yeah. So 4 years UWA. Then I took a year off and just sort of stuffed around a bit, I suppose. I've been doing that. I applied for a position with Australian Volunteers Abroad, which is like a overseas service bureau. So you go overseas and you get sent to you can't call it a third world country anymore. You call it the southern rather than northern countries. And I ended up in Sabah in Borneo and worked with the Department of Veterinary Services there as a pasture agronomist, which really meant supporting the senior agronomist doing stuff. But it's where I got a bit of a love for doing things a bit differently. So I did nutrition experiments. I did variety evaluations for disease tolerance. I did my first herbicide experiments there because we never touched on herbicides much doing the undergraduate course.
What year was that?
That was 79 and 80. Mm-hmm. And so we had giant mimosa. Mm-hmm. It's absolutely one of the most vicious weeds you can imagine. It has thorns all over it, and if you get them in your skin, you end up with infections really, really easily. Anyway, this stuff grows up to 3, 4, 5 metres high in thickets like you can't imagine. Seeds are extremely long-lived in the soil. And, of course, we were trying to grow pasture for cattle. And once the thickets started, the cattle wouldn't go into it. They just avoid it. So I did my first chemical experiments with Agent Orange. And true. I mean, it was post-Vietnam War, drums of Agent Orange, which
Left
ended
over.
up in Sabah. And I'm sitting there thinking, okay, what herbicide am I going to use? Oh, we've got this stuff in an orange drum over the back there. I said, do you know what's in it? But we think it should kill it. It did. It killed it absolutely stone dead.
Any bad side effects from using it?
Look, I only sprayed it a couple of times, but I never saw any effects that I could speak of. I mean, I used a little bit of protective gear, but not a huge amount.
Yeah.
So, yeah, that was sort of one of my first weeds experiments, I suppose, that I did.
And then you became one of WA's weed experts.
Well,
I would
possibly.
say that. I would say that, yeah.
I mean, Sabah was really interesting before I became the weed expert. I mean, I did the thing that I really remember was my stuff showing sulfur deficiency in grasses there. Because, we used to put nitrogen fertilizer on. Why aren't our grasses growing?
They're not getting green. They're still yellow.
Getting green. They stayed yellow. Oh, there must be something wrong. So we tried everything. Finally, one day I said, right, I'm going to use the shotgun approach. So I just grabbed everything I could out of the shed and I put strips down all over the place. And after a week, that strip's green.
Was that one?
3 weeks, that strip's green. After 5 weeks, 6 weeks, and that strip's green. Hang on. Why are the ones all showing up green? All of had sulfur. Readily
Quick release,
palatable,
mid-release.
and slow release. And you sort of sat there and thought, So I did a whole study on it and talking about how much sulfur was coming in on the rain and what the soil types were like. And we did the soil analysis. I basically wrote a report that said, well, if you want to grow grasses in Sabah where it rains a lot and you've got these absolutely horrible white, highly leached sands, you have to put sulfur. That's what I thought was like, it was interesting doing the work on mimosa, but the sulfur was just something I'd never done before. So that was really good.
I found the same thing in Congo where I grew maize with good dose, 200 kilos of DAP.
Yep.
And they looked great, but in these patches of soil, which were lighter, they went yellow. So I was desperately trying to get hold of some potassium sulfate because I knew it was one or the other or both, and couldn't end up getting any, unfortunately.
So I came back and anyway, there weren't many jobs in the ag department, but I went to uni and they said, oh, well, we've got some PhD. Do you want to apply for one? Just by chance, there was one that talked about herbicide tolerance in wheat, put out by one of the wheat councils, the state wheat council, I think it was.
And
so
Marcus Blacklow?
So, I got it, and it was for genetic and environment. So Marcus and Roger Boyd were my two supervisors. Now, I mean, in my fourth year honours project, I'd done genetics and canola. So I already had a very strong, predisposition to doing some genetic stuff. So, I did 3 years with Marcus and Roger, and it was absolutely by chance that at that time we had Glean come on the market, so-called sulfuron, and we had the first reports of myling wheat being damaged by Glean. So I tried a couple of different herbicide things and they didn't seem to work. But when I heard this stuff, I thought, okay, well, we'll give this one a go. And it took me, like most PhDs, you sort of sit there and think, oh, yeah, you'll sort it out in the first week. Well, after 12 months, I'm sitting there thinking, I haven't made any progress. What am I going to do? I'm supposed to have done something. So I thought, okay, well, we'll change tack a bit here. And the next 6 months, I worked out a technique for how to study chlorsulfuron in the glasshouse and went back to basic physiology and applied basic plant physiology to the whole process. And from that point onwards, most things fell into place. So I did some field experiments, lots of glasshouse experiments, pretty much cracked the whole thing open.
Well, it was a good work. I remember reading a lot of your stuff, and interestingly, to link it back to Four Farmers, the company that's hosting the podcast, that's what got Four Farmers going, was a Glean coming off patent and Four Farmers halving the price. So farmers, love Four Farmers for that.
Well, I certainly did do a lot of work in the field with Glean, lots and lots of herbicide tolerance experiments, looking at, yield reductions anywhere from 5 to 20%. Back in the day, Cule and wheat came on and it was going to revolutionise yields, and it did. Unfortunately, it was highly susceptible to herbicides, not just Glean, but also to anoxy herbicides. All the heads used to drop off if you put it on at the wrong time. And it turns out that I think the gene for both of them is slightly related as well.
Okay, so now you've had a wonderful career in the Department of Agriculture, and
I did a lot more than that.
It did, yeah.
Too.
Yeah. Well, share some of those exciting things.
I mean, I went on to do the herbicide tolerance work. Then I became the project manager, integrated weed management, where I ended up with pretty much most of the weed management team there. Did that for 5 years, and then I thought, am I going to stick around here forever? No. And by chance, the job came up for program manager, pulse and oilseeds. And I thought, well, I'll throw my hat in the ring. You never know, I might. Of course, I didn't get it, did I? Until 2 weeks later, they said, the top candidate has pulled out. It's yours. I thought, oh. So I did that for basically 12 months, became program manager of pulse and oilseeds. And that was a pretty steep learning curve, because suddenly I went from a little project to quite a big project with lots of staff. But I wasn't actually responsible for them. This was really interesting, because our structure then was a funder purchaser provider model. And, my job was to be the purchaser, as it were, of the services provided by the research staff. So you get to look over all of the projects, but you didn't actually have to worry too much about the stuff, but you had to set strategic direction and things like that. So did that for 12 months. And then the Director General of the day said, right, we've got a shortage of money. We're going to start merging things together. So suddenly
went back to the way it was before.
Well, no, it didn't go back to the way it was. Suddenly, not just pulse and oilseeds, on grains side, got cereals as well. Steve Trevannon used to be the cereals program manager, and Steve was sort of, I suppose you could argue, was moved a bit sideways, quite a lot sideways. They gave me all of grains to look after, which was a pretty big ask, because it was by far the single biggest dollar spend in the organisation at that time, probably, apart from the APB.
So what do you think through your career was one of the biggest changes that you've experienced in agriculture?
Well, I suppose that change in terms of where you always, within the ag department, was you're with a small team of people where you got on really well with, you learnt from everybody else, to now this model which was a bit more dispersed, where people were put into project teams rather than a disciplined team, whereas the disciplines were really, really highly focused. So you lost some of that discipline integrity, I think, was one of the things that concerned me most. But, you're looking at lots of things happening in parallel through this whole system. So, yes, you're getting a change in the organisational structure within a government agency. But at the same time, you've got this very big change that's happening in agriculture in general. And so, from when I joined, the fact that we had the first of the in-crop selective herbicides like Hograss and Glean and Logran, and since then a whole range of other ones have come along, has actually probably done almost the biggest transformation and allowed us to pretty much manage
weeds very well.
We were relying upon cultivation, with all its associated problems, of which wind erosion and various other things, loss of organic matter, major. As soon as you swapped out of not having to cultivate, and that was augmented with having glyphosate and paraquat and dicot, which meant you no longer had to cultivate. You could actually do the whole lot, knock down as well as in crop at the same time. You actually did a biggest transformation. Now, you can argue and say, well, we've got modern fertilisers, but you could use those. We were using those fertilisers in the old system. We just didn't have the weed control.
Well, Bill Bowden, who I just recently interviewed in the last podcast, he said, that's one of the biggest changes he's noticed is he used to drive through the wheat belt and there'd be weeds popping up all throughout the crop like a dirty haircut, And he said now you struggle to find one, which is testament to the herbicide technology. Not everyone likes herbicides, David. So maybe address that.
Well, it's the clean crops.
Be very sympathetic to people that are from the city that don't have any idea what we're talking about. And what herbicides are chemical? Aren't chemicals all bad?
Oh, you're made up of chemicals, aren't you, Bill? So you must be bad.
Well, that's true, and I'd acknowledge that. I am bad at times.
Look, there are good natural chemicals. There are bad natural chemicals. There are good.
Give me some example of some bad natural chemicals.
So if you look at ARGT, the toxin that's in ARGT,
Annual ryegrass toxicity,
is probably the fourth most toxic compound that we've got around. And it kills sheep like you wouldn't believe, and cows and horses. And, I can still remember my sheep dying from ryegrass toxicity.
They don't need to eat many, do they? Only.
Oh, look, I thought there was no ryegrass in the paddock, and yet suddenly I've got 20 dead lambs. And my neighbours are the same. We'd, put the sheep in and mix it. These are highly potent toxins we're talking about, and people seem to think that, all nature is soft. Well, it's not. It's really, really nasty sometimes. So you see,
that's a biological agent.
It's a biological.
Can you tell me a chemical, natural chemical that is dangerous, an organic sort of?
Well, that is a natural chemical.
Yeah.
I mean, it's
it's biological, they're not.
Well, no, it's a chemical. It's
a chemical made by the biology, yeah.
It's chemical, right?
Thank you.
So you just got to discriminate. And I'm told it can be pretty good biotoxin if it was to be manufactured synthetically. So, except for you can't make it.
Okay.
Fortunately.
Yeah.
Look, there are all those things with all the gradation of different things. So, you look at the pyrethrins that come from chrysanthemums. They're the forerunner to modern synthetic pyrethroids, generally extremely safe.
Which is what we put in a mortine.
That's mortine. Well, these days most mortine is made with a synthetic pyrethroid. It's not with the natural. But you can still buy the natural ones. They're expensive because you've got to harvest millions and millions of pyrethrin flowers to make a very small amount of natural pyrethrin. But yeah, it's relatively, well, when I say relatively safe, it's about the same toxicity as common salt.
So why would people think that if man's played with it, it's evil, and if man hasn't played with it, it's not evil? Why?
Oh, look,
what's going on there?
I think people get a perception from everything they read. You can go back to things like books that were written, Silent Spring with Rachel Carson, where you've drawn conclusions about things based upon often, non-experimental. It's epidemiological. People get a view of the world about those things. And look, legitimately, there's no doubt that some of these things did have effects. It's pretty well documented that you can get eggshell breakage as a result of DDT and all those sorts of things. But, doesn't mean that therefore it's going to, ruin the whole world. Well, it breaks down relatively quickly in the soil. People who were exposed to DDT in the chemical manufacturing facilities never had any higher rates of birth defects, deformities, or cancer than the general population.
That wouldn't be widely known, I don't think.
They're not very widely known at all. The studies were done on the people who were in the DDT manufacturing plants over a long time.
Well, I can add to this a little bit because we had Dr. Nigel Wilhelm on one of my study tours that we took to America. Yeah. And on one of the moments he said, let me tell you a document that never got released. Have you seen that document?
I've never seen that.
Well, what he's saying was there was two villages. Both had 6,000 people. Socioeconomically, they were similar. They were rural USA. They both had a river running through it. Both had fish in the river. One place had the DDT manufacturing plant up the river about 5 miles, and the other one didn't have one. And they looked at every aspect of human health you could think of, everything. And there was no difference between any of them except for one thing. One group lived 5.5 years longer than the other group. And surprisingly, it was the group that had the DDT factory up the river. Now, you don't hear that. And but you've heard it.
Well, I've heard different stories, right? The studies that were done in the Philippines on the people who were in the DDT manufacturing had lower rates of cancer. And the reason they think is when they studied the liver, the enzymes were upregulated in the liver. That helped actually control other things in the body. Now, that's just DDT. There are other, obviously, what do they call them? Chlorinated
CFCs?
Well, they're not CFCs, but they're basically chlorinated chemicals like DDT, etc., a whole range of other ones like DDT.
Heptachlor.
And some of them heptachlor, which do have more serious effects.
Yes.
And it's like everything, different levels of toxicity, different levels of doses, and so therefore you end up with different effects.
Yeah. So these herbicides have been wonderful for West Australian agriculture. Herbicides now, I'm not talking about we're going away from DDT. In terms of hygiene and yields, we're seeing tremendous yields with clean crops. And you had a big part in that. So, but we got resistance to some of these herbicides too. But you can go there now or you can go there later. Well, you've got something else in your mind you want to get out?
I think herbicides in general have, probably, and people have commented that, the amount of labour saving that's been done globally has been phenomenal. It used to be that most people spent the majority of their time weeding paddocks, particularly in lower socioeconomic countries. We didn't do as much. We just lived with, wild oats growing in our wheat crop and, we got lower yields as a consequence. But yeah, it's actually been probably one of the biggest side effects or main effects of pesticides. You save labour. People can go and do other things. They can earn income somewhere else. And very few people actually recognise that. They just think it's, it's killed a few weeds. Well, there are flow-on effects. People aren't aware of the flow-on effects.
Yeah,
well, there are herbicides which have potentially much bigger effects. You look at paraquat; it is quite toxic if you drink it or you spill it on your back and you don't wash yourself. Yes, and we know that people do get paraquat poisoning around the world. But relatively speaking, it's actually way, way, way less toxic than in smoking.
Well, people see it on the TV or when people are anti-chemicals, they have an aeroplane going over a field and spraying out all this chemical and it's flooding everything. But the reality is 90+% of that is water.
Exactly. Well, in the worst case example, it's probably 5 or 10% is chemical. But yeah, you're right. The majority of it is water, but of course it's got the chemical incorporated in it. And as I said, there are different levels of toxicity. The other thing, of course, is these days we mix fertilizer with the chemical. We sometimes put it out at, very low volumes, 5 or 10 litres per hectare rather than the 500 or 50 litres per hectare. So there's a whole swag of things that have actually changed in that space. I mean, I think the important bit is the selectivity that has changed. The fact that you can actually, it's a bit like taking an antibiotic, right? And people don't realise that it's exactly the same process. The antibiotic kills the bacteria but doesn't kill your body. In the case of herbicides, it kills the weed, doesn't kill the crop. It's exactly the same sort of process is occurring. And as you just mentioned with resistance, it's the same sort of process. One bacteria survives the antibiotic, you get a drug-resistant bacteria, which is becoming a bigger and bigger global problem. Well, in the same way, get a weed that survives, it becomes resistant, multiplies, and within a few years, if you keep using the same herbicide, you've got herbicide resistance.
I remember 25 years ago, I had a chat to you about multiple groups together. And you said to me one day, Bill, herbicide resistance is just not a big deal. All you have to do is put 3 modes of action together in the one hit. And what chance has anything got of surviving those 3 groups?
Well, generally that principle works. Where it fails is if you have a metabolic pathway that can deal with both of them. Yep. And there are some metabolic pathways that can deal with lots of herbicides at the same time. It's unusual, very, very unusual.
Or if you've got a history of all 3 of those herbicides being used in the past, now you put them together, they won't work quite as well as
they can fail often much more quickly.
Yeah.
So the general principles, it doesn't matter whether it's weeds or bacteria or fungi in crops or whatever it is, the general principles still apply for resistance. It's natural selection in a way, actually just being sped up many, many, many times. So I can still remember when Roundup came into Western Australia because my dad went and bought some, and he walks in with this one little
1970 something,
about 72, 73, I think it was, because he comes in with this little bottle of he said, $100 for a litre. This stuff is like gold. Don't waste it. And of course, we only wanted it for the orchard, just for a patch of soursop, because we couldn't kill soursop. 30 years of ploughing had not got rid of the soursop. So he said, right, we're gonna blitz it. And it did. Worked absolutely brilliantly. And, I hadn't even gone to university then, and I'm still seeing, besides. And we've transitioned now to the point, where we can put Roundup, glyphosate into crops and leave the crop behind. Now that has been something that, has been a transformation that's even more relevant.
Yeah, and there's some people are scared about that technology. The funny thing is, when I was in Africa, and I'm not scared about it because I understand the science to a reasonable degree. But when I was in Africa, they said the reason why the West doesn't have any children is because you use GMO crops. Of course, could nothing be further from the truth. So sad, so Africa's being kept poor by not being able to use this technology. And I remember, David, I was sitting on my bum one day with 9 other workers, and we hand-pulled weeds on 1 hectare. And it cost me $3 American for each worker, so $30, say, and we only did about a quarter of a hectare, and we had to do it again. We had to do another 3 days, and then 3 weeks later we had to do it again. So here we spent $200 a hectare, 300 Australian dollars, to clear the weeds from our maize crop that went 3 or 4 tonne. It should have gone 6 or 7. But if we'd put Roundup Ready technology in there, I had this moment where I actually teared up, because I just sold my farm at Morrowa, and it started raining on my farm since I left my farm, so that made me feel a bit sad. But I had this rogator that would spray 120 feet, and I'd do it. I was a bit cheeky. I'd go to 28 kilometres an hour, so I could knock off 500 hectares in one day just going back and forth. So here's 500 hectares with every weed being blitzed, and I know it. Every weed would be being blitzed, bar the 1 or 2 that for whatever reason I missed, but virtually every weed. And I compare it to what I'm doing, which we're not getting them all, because a lot of them got, bulbs underneath, and so they just regrow. And so I had this moment, what is Africa doing? You get spending all this time pulling out weeds when you could be making good crops, good temperature, good soil, not a lot of P, but 20 parts per million is pretty good. 5 parts per million sulfur, you'd rather have 8. 60 potassium, 80 is better. So you've got this soil that's not bad. It's not far off being okay, but not doing it.
So, I'm sure Africa will come to its senses one day. South America has.
Brazil.
Much of Asia is doing the same sort of thing. So Africa will catch up slowly. But the ability to selectively take weeds out has been something that's been really interesting. And the first time I ever actually did it was in my veggie garden at home when I was 15.
Got some Fusilade, did you?
No. Grass. Long before grass selectives came on the market, I used to have portulaca, pigweed.
Oh, yeah.
Never die. And it used to absolutely go ballistic over summer in the veggie garden. And I'd spend hours weeding it, hand weeding it out and hoeing it. And I eventually got to the point where I said to myself, I can't do this anymore. And I went to one of the hardware shops and I looked at everything. And sure enough, there was one chemical there that you plant everything and you spray it on the soil surface and it stops the weeds from germinating. It just makes a layer. And I thought, I'm going to try this. And I did. And it works an absolute treat for one species in particular, and that's pigweed. Never die.
And the herbicide was?
I can't remember.
Wasn't atrazine?
No. It's endothal or something like that, and it worked an absolute treat.
Yeah.
But as soon as you disturb the soil, it stops working, and up comes all the pigweed.
Yeah.
So you've got to leave everything intact. You just leave it there, and as long as your crops have come up through it, then you spray it on, you get this really good control. And that was my first experience of selective weed control, but in my veggie garden.
Yeah, okay. Now, so let's go back to GM crops, because I know you've got pretty good knowledge of glyphosate. And of course, in America, Bobby Kennedy's in there, and he, some things that Bobby says I think are really good. They've got to look at the obesity. They've got to look at, there are issues, autism and all of that. Well, it's good to look at. Science is never settled, as we should know, because we're scientists and we've criticised each other's science and we've sharpened each other on occasions. So, but I'm fully with you on this one. He's had a court case in California where a gardener for 2 years was having glyphosate fall down the back of his back, and this guy supposedly got non-Hodgkin's lymphoma from that. And he won the court case. $270 million was awarded. A few months later, it got appealed, became $80 million. A few months later again, it became 20-something million. And now the guy's not dying. He's still fine, and he's got his money, and Bobby's now in a very important position in the US. Yeah. So what would you say to Bobby Kennedy about glyphosate and non-Hodgkin's lymphoma, given the court case we had in Australia just last year, where the judge said on the basis of the evidence he can see, there's not evidence to support the idea?
I mean, the challenge always is it's circumstantial. And, you've got to look at all of the other evidence that comes from animal studies and other ways. Unless there is some other justifiable reason, a judge, and I can't predict what any judge would do, a judge should look at the evidence rather than necessarily look at the fact that, one person says this and therefore I believe that person over all the other evidence.
And I think that's why they had it in California, because actually the jury was the ones that decide, right. And if you get them crying, the evidence was thin. So, so the mechanisms, you've probably read the same scientific papers that I've read by Eric Giles Gillies and Judy Carman, the rat studies that are pretty poor science.
They're not well conducted, let's put it this way. Having appropriate level of controls, being able to compare to other products of similar nature, all those sorts of things become really, really important if you're going to do these sorts of studies. And you actually got to look at the physiology of it. What does the physiology say when this product is put into any animal model? And humans are animal models, just like, what's that? Slightly different. In general, we tend to have less effects, still an animal model. So you got to look at, the whole range of those sorts of things. And my personal view is it's not a lot of evidence to support that occurring. I mean, just simply because, first of all, we don't have enzymes that glyphosate blocks. The one thing people have said, what happens, of course, is it complexes the copper and the zinc and other trace elements in your body. You become deficient, and so therefore that's why you might develop carcinomas or anything else. But again, not a lot of evidence to support.
And it's the dose that makes the poison. And when you think about when we apply it with wheat, it's before the crop's even planted, glyphosate breaks down to NADP or something, which is, it's
ultimately, it ends up just being incorporated into bacterial bodies and becomes part of their phosphate and nitrates in the system. So, pretty much everything that we do goes back into bacterial bodies. Doesn't matter whether it's a human or a chemical. That's what happens. It just takes a little bit longer with some, a bit faster with others.
And, how much of it,
How much? Absolutely.
I still use sugar in my coffee. I know I probably shouldn't, but I just don't use a lot, David. Look, the doctor said, my GP said yesterday, in good numbers for diabetes.
Thank God for that.
Yeah.
One of the really fascinating things is very few people really read the scientific literature ruthlessly and religiously. I've always read scientific literature.
You're a geek in that regard. And that's why I enjoy talking to you.
I discovered a paper written back in probably mid-80s where a scientist in, I'm pretty sure it was Slovakia or somewhere like that, he took bacteria out of the soil, one of the fungi, and he put it into a vat and produced glyphosate. And you sit there and say to yourself, why would any fungi use glyphosate? Well, guess what? It's a perfect protection agent against other fungi and bacteria, right? It blocks the same pathways in other bacteria. So some of these fungi are actually producing as a defense mechanism against other species.
So glyphosate is an organic herbicide.
So it's actually a naturally occurring compound. And then you sit there and ask yourself the question, well, why is it that when they took the enzymes out of bacteria to put into plants for genetic modification, what were they doing? They were actually taking it out of the species which were defending themselves against the other species which were producing the glyphosate. So you actually got this, war going on in the soil between the glyphosate producers and the non-producers who are defending themselves with different range of enzymes, either blocking the pathway or metabolizing it. And so you could actually take it out and use it. And if you actually go and spray glyphosate onto a lot of moss species, it doesn't kill them. You can pour it onto moss, and the moss just quite happily just keep growing. I do it on my fire breaks all the time, and I've now got a fire break which is made up of 100% of moss, because when I put glyphosate on it, it doesn't kill them. So they've already got a natural mechanism by which they can actually defend themselves.
Well, and going back to herbicide-resistant ryegrass, for
Its
example.
natural resistance is already present in some species.
So those genes that are in the ryegrass that resist the glyphosate are the genes that end up going into the GMO crops, the soy.
Not necessarily.
Or similar mechanisms.
Similar, yeah. Some of them are breakdown, some of them are actually site of action.
Yeah. So if people are worried that this is not a natural background, it basically is, really.
Yeah. So and it's pretty much what's happened. You've taken a naturally occurring gene, put it into a plant, and modified it. And people think it's not that, but the whole GM debate got very worked up at one stage. I was the department spokesman for the GM crops when they were being released, and there were lots of arguments. We're going to have Roundup Ready canola. It'll become the major weed along roadside verges. What? That's all we'll see is yellow flowers everywhere. And
20
now got Roundup Ready canola growing over 95% of the canola area, and you drive out there in springtime and you virtually do not see a yellow flower on the roadside verges. So that was, these sorts of arguments that were made. You can always have mishaps. There's no doubt about that. And every time you make a GM plant, you can end up with strange effects. I worked with John Hamlin to make glyphosate-resistant lupins back in CLIMA, Centre for Legumes and Mediterranean Agriculture. And we thought this was going to be a really game changer for lupins. And it would have been, but of course, we couldn't get it through the AP, through the Office of Gene Technology regulator as it was then. And it worked brilliantly. Absolutely. You could take ryegrass out at flowering time and hardly touch the lupins at all, and yet the ryegrass all had 100% seed set. Great idea. What was interesting was looking at the plants that were created, you had all sorts of variations, because when the gene gets put into the plant, it's not going in any one set place. It would go in all sorts of places. We'd upset flowering time, leaf shape, structure, all sorts of variations. And so you had to screen it out till you found the few that weren't being affected. So like anything, gene insertion and gene regulation can be upset by the whole process.
So most people don't realise that if you have GM crop sort of like Roundup Ready, that you're actually putting less herbicides on. They think you're putting more and you're flooding it. Yeah. But you and I know that there's 6 or 7 herbicides you might want to use, and you probably still should use some because you want to protect the glyphosate.
Usually replacement is the thing that were occurring. So we'd take an atrazine out of the system and put glyphosate in most cases. That's been the single biggest change.
And atrazine's banned in Europe.
Well,
because it gets...
But not in the United States.
Yeah.
So you
can get in water table.
Is it really as nasty as what everybody says?
Probably not.
Probably not. But
certainly glyphosate's safer.
The reality is in those places where your water supply comes from the ground, you try to minimise pesticides, you try to minimise nitrates, you try to minimise phosphates, you try to minimise those things which might or sometimes do have effects. And blue baby syndrome from taking nitrates is well recognised for grass. But, so is arsenic. Arsenic in groundwater is a huge problem in India and Bangladesh, where they
Argentina.
In the United States, arsenic in groundwater is becoming an emerging problem as they deplete groundwater.
And it's just a natural product that's in the groundwater.
Well, it's a natural, but it's toxic. That's the point of it, Yeah. So you've got to look at the contrasting evidence for those sorts of things.
Lindsay Chappell told me he went to Bordeaux, and he said he sent me a message from southern France. He said, "Bill, you wouldn't believe it. The organic farmers can't grow cows on their grass anymore because the soil's too toxic from the Bordeaux mixture, which is copper sulfate. Excessive use over a long period of time. Organic."
Well, let's put it this way. It's only organic in the sense that there was a certain year at which everything was decided we will no longer use superphosphate in the system. And it goes back to about 1923. And anything that was used prior to that was deemed to be safe. copper-based fungicides, arsenic-based fungicides were deemed to be safe. No evidence pointed out, but they were deemed to be safe, so therefore they were allowed to be classified as organic. Subsequent to that, anything like superphosphate and all the other things were deemed not to be safe, so therefore you didn't do it. So it's really a delineation that's a bit of a, say, false one, but it was done on the basis of a group of people who decided this is the way we're going to do it. And if you look at copper and zinc, which have been widely used as fungicides, copper toxicity is huge if you use copper-based fungicides on your fruit trees. And so we've got examples in New Zealand, apricot orchards completely dying from copper toxicity. Oh, yeah, it's a big problem. Zinc toxicity.
And it's hard to get them out, the heavy metals.
It's almost impossible. You've got to remove the entire topsoil layer down to about 20 centimetres in order to amelioate it.
What do you do with that?
You've got to go and dump it.
Where do you dump it? Solution to pollution is dilution.
Well, mostly it goes into landfill underneath houses. That's often what we've done with it. And you look at all the housing estates around the world that are sitting on toxic waste pits, and there's a lot of them. So, yeah, look, you can condemn it and say, well, it's organic, but it's toxic. Yes, it is. But the reality is we still use copper-based fungicides hugely. But it's nice to think that we've also got all these molecules which are synthetically made, which break down to nothing more than water, carbon dioxide, some nitrates and phosphates, which actually go to make up the chemical bodies of bacteria. And, I've been involved in a couple of major research projects in my time, one of which has had major international ramifications, which I don't talk about much, and most people aren't even aware of.
Oh.
And that was the development of imidazolinone-tolerant crops. So the IMI-tolerant wheats that are used in Australia,
Clearfield,
the Clearfield wheats, and not so much the barley and oats, but the Clearfield wheats, I was very heavily involved in the development of those. When American Cyanamid came to us and said, look, we've got this variety which we think might be useful in Western Australia. What do you think about it? And I can still remember the conversation I had with Mike Jackson from American Cyanamid out in the paddock looking at brome grass growing across a wheat crop. Do you think it would be useful if we had this product that could take brome grass out of wheat?
You betcha.
Thinking, oh, have you really got one? Yes, we do. And we've got the gene too. At that point in time, it really sparked up my interest. And I said, well, okay, let's get a project going. So for 10 years, we ran a project within the department of developing IMI-tolerant wheats with the single gene. And, we got the first variety out there, J and Z, and two other varieties that had the single gene in, but they were never perfect. And you could often get crop damage. Suggested to the plant breeders, we need a second gene. And so with Ian Barclay and Kevin Jose, we set out to produce the second gene. We used mutagenesis of wheat using sodium azide, and we screened a ton of wheat. Put it through mutagenesis, then we screened the population for M1, M2 populations. We got 3 plants which were resistant. And of those 3 plants, 2 of them had a different gene to the one that was originally produced by American Cyanamid. We crossed them together and produced a 2-gene model, which gives you almost complete resistance to imidazolinone herbicides. And that second gene, which is the one that Kevin and Ian and I have a patent on, is the one that went global.
So you're a rich man.
No, I don't get a single cent.
Why not? What?
Because it was done in the Department of Agriculture, and government employees are not liable but able to receive benefit from the work that they do as part of their normal.
You could be so rich and exceedingly arrogant, David.
I don't know about so rich. Anyway,
well, I went to Eyre Peninsula and I witnessed this transformation just this year because they weren't strong on disc seeders in South Australia originally. We were because we had acid soils, but you had nematodes and you had rhizoctonia and you had brome grass. But now with the IMIs, they've got their rotations stronger, particularly on the Eyre Peninsula. It's very good. There's a large proportion of the farmers using disc seeders with no soil disturbance, and they don't have ryegrass, so they don't need to use trifluralin. So that sort of made me think, IMI, very clever molecule. It breaks down in alkaline soils, as you and I know. So Yeah. No,
So its bacterial degradation is very strong. So as long as you've got wet, alkaline soils, it breaks down really fast. The beauty of it is it gives you residual activity depending upon which combination you use. Extremely high safety levels to animals and earthworms and humans and stuff like that. So
very low dose.
and low dose. I mean, we're talking about sometimes teaspoons per hectare.
Yeah, and a hectare is 100 metres by 100 metres.
10,000 square metres.
Yeah, a teaspoon in 100 metres by 100 metres is all you use for one molecule particularly, but double that or triple that, but still not much.
But I've done a few of those. I mean, I was probably the first person to advocate using metribuzin brodal for radish management in lupins. And I can still remember quite clearly the day where the farmer rings me up and said, I've got all this huge radish and nobody can tell me how I'm going to get rid of the radish out of my lupins. And if I don't do something, I'm not going to have a lupin. So I suggested, well, try 100 grams of metribuzin with 100 mils of brodal. Anyway, he did.
He rang up a week later.
3 weeks later, he rings up, you've killed me crop.
Have another look.
Oh, my God.
So you went back.
Am I going to get sued here?
Was this up the Geraldton area?
I was starting to get really, really worried. This is, east of Geraldton.
Yeah. Being
3 weeks later, he rings up. He said, I have never, ever seen such a response. My lupins are fully out in leaf and flowering again, and the radish is dead.
Yeah.
I want you to come to a field day.
Wow.
Talk about it.
Yeah.
So I went to the field day, and sure enough, and I'm thinking, he's done 500 hectares, He did 5 hectares. And here's me stressing my poor little heart out.
You got to get sued.
I'm going to get sued for killing his crop. And he said, this is just brilliant. Anyway, I thought, okay, so we now got a combination that'll control radish in lupins, but we don't know about the lupin response. So I started doing the herbicide tolerance work on lupins, and lo and behold, aren't we lucky that we had gungaroo lupins with high levels of tolerance to metribuzin.
Yep.
Wow. Suddenly, within 2 years, we were able to pretty much take it out there. And while it didn't have a label, both metribuzin and brodal were both registered on lupins, so it gave us the shoe-in to actually create the label and get the companies to create the label. So we were very lucky because DuPont were actually prepared to go away and do it.
Yeah.
If they hadn't done it, then it probably would have still been sitting there.
Now, you're a bit of a breeder as well. Tinker away. You've made us a canola variety, and you've seen your little hill plots with wheat and all sorts of interesting things. But if I could steer you to lupins really haven't, they've struggled. Even though we've got acid sandy soil suited to cool season climate, the yields, people would say, first year I grew lupins, I grew 3 ton per hectare, I was up to my chin, what's that, yarry or something? Yeah. I've heard that story a dozen times in my career.
A few people do that.
And they struggle to get 3 ton now, and even though we've shortened it down to our knee height. So what do you think's going on there? Why can't lupins consistently get us up on a high yield area?
Well, I think there's probably 3 reasons. I mean, one is they do seem to accumulate disease organisms in the soil a little bit better. And it makes sense because they're producing a lot of nitrogen-rich material, which goes into the soil.
So there's a honeymoon period with Ilyari.
Well, I think
that sort of thing.
I think the first year on virgin soil, effectively, is it gets a bit of that. The other thing is we nearly always inoculate them with a really, really good rhizobia the first year. Subsequent years, we tend to think, there'll be enough bacteria in the soil so we don't need to worry about it. So there's a little bit of that occurring as well. I think those 2 things. And often it was the case that the first year, the weed levels were lower. And so because we were coming out of long-term wheat, barley sort of rotations, whereas by the time you get into the 3rd, 4th, 5th year of lupins, your weeds are starting to build up and starting to compete. So you've got a number of those things. To one extent, probably the first lupin crops that were planted often were planted with superphosphate, single super, rather than now the more
MAP.
Well, MAP or triple super, which didn't have any sulfur in it. And so I think you changed around some of that. Some of the superphosphate, when it was used, often had trace elements built into it, the copper, zinc, molybdenum. And molybdenum is very important for bacteria nodulation. So we probably have thought, we can just keep doing this, forgetting about all the basics that actually go along. The other thing I think that's happened, which is probably a little bit controversial, is that we actually had a run of, back in the 80s and into the 90s, of actually quite good seasons. We had really good rainfall, consistent over the whole life of the year. Plants were able to recover from stress. And then by the late 80s, we were starting to put more and more diflufenic and brodal into the system, and it can be pretty toxic, as can Eclipse, to lupins, and we were probably starting to suppress our lupins with herbicides. Doesn't sound like much, but it's just sufficient to actually tip them over the edge on some of these things.
But if you think of maize, in America going from 1 tonne to 15 tonnes per hectare, and wheat going from half a tonne to 3 or 4 tonne, depending on your rainfall, or 5, 6 tonne, lupins just haven't really. They seem to start off at 2 or 3 tonne, and they're sort of still that, and their price, of course, is not high.
If you look at the amount of breeding work that's been done, at most, it's 50 years. And it's been up to one person, John Gladstone's, and then 1 or 2 other people who came along, right? Mm. If you look at maize, it probably has been running in one way or another for breeding since Mesoamerica of probably close on 500 years. Mm. If you look at wheat with natural selections and all of the other things and the diversity that it's had, which we've been able to access, it's 10,000 years.
Yeah, and it's every country as well, whereas lupins is pretty much just us.
Narrow-leaf lupins, it's only really Western Australia. So you're actually asking a lot of a new crop to suddenly perform in the same way an old crop does. Also, the other thing about lupins, of course, is if you look at Western Australian soils, they're pretty bloody depleted relative to the rest of the world. There's a lot of variation. Even within paddocks, it's huge amounts
depleted,
of
you mean its native
state is depleted because they had 4 billion years of,
perhaps,
probably 3.5 billion years of not being under the ocean.
Perhaps.
No, not perhaps. It's a fact, Bill.
There was a tsunami in West Australia 5,000 years ago, Steve.
Yeah, but that's different. That only happened on the coastal plain, Bill.
Oh, it depends what you believe.
It's only on the coastal plain, Bill. The evidence is there. So you've got to sort of sit there and say, lupins are it's a pretty hard ask. But people are starting to consistently get 3.5, 4 tonne yields with the new varieties and by putting good practice back in place with better herbicides, with better fertiliser strategies.
Yeah.
Personally, I think, 4 tonnes per hectare is starting to be achieved. And it'll actually be, I think, done on some of the better sandplain soils and also some of the good red loam soils where they're deep. I think that's the secret. If lupins can get to 2 metres, this is my personal
roots.
2 metres, the root gets
soil.
2 metres, they'll do really well. It's when they hit that shallow hard pan at 30, 40 centimetres, that's what's...
Yeah, well, 2/3 of the state's got clay at a depth of 20 to 30 to 40 centimetres.
pH, as soon as they hit a pH which is above 6.5, they just don't take up the nutrients in the same way.
Okay, and so what other breeding schemes have you had that you've really enjoyed?
Really want to know?
Well, give us your...
Well, Tranby canola was the variety I developed as a result of playing around in the glasshouse with, I think I had the officer in charge at Katanning Research, Katanning.
Joe Burtis?
No, it wasn't Joe.
Ian Mailing?
Not Ian Mailing. It came after.
Yeah, lost touch. Yeah,
I know it was around Ian's time, Anyway, he said one day, this is what I want. Do you know anybody who can do it? I said, what do you mean? Well, we've got all this background material that was developed by Narendra Roy, and it's all sitting in a warehouse somewhere, and we've never bothered to use it, and it's got some really, really excellent blackleg resistance in there, absolutely brilliant blackleg resistance.
Mm-hmm.
What do you think you could do with it? Think somebody could do it? I said, give me So I just did it in the glass house, all right? Nothing out in the field. All I did was cross a highly blackleg resistant variety with a very early maturity with a triazine resistant material and selected out all these plants, tested them out in the field and pretty good. But their levels of glucosinolate were too high. And by chance, the plant breeders from Salisbury, I think it was, from New South Wales Ag was over one day and he was looking at it all and he said, yeah, it's pretty promising stuff there, but you got too much glucosinolate. Use this variety as your other parent. Said this is a New South Wales variety they just released, a very low glucosinolate and low erucic acid. And he said, use that as your other parent. So I crossed it into one of the better lines. And within 4 years, basically had developed this really early maturity, triazine-resistant line. Wasn't fantastic blackleg. Yielded really well.
I remember John Moore saying to me, why would farmers want to spray a crop with atrazine that's 30% lower yielding than an open pollinated non-herbicide tolerant. And of course, you and I know that weed control is pretty important. You can lose half your yield if you don't control your weeds.
Well, it turned out not to be lower yielding, that much lower yielding. When we stacked up Tranby against the best early flowering.
I was thinking of Karoo before that.
Oh, yeah, Karoo. Yeah. When we stacked it up against Karoo, it actually wasn't lower yielding.
No.
And the few farmers who I know who did use it out on the eastern wheat belts said, like, this was just a game changer. Because they said, early flowering in our dry, short season environment. Anyway, I couldn't really compete because then Trilogy came along and a few other ones came along.
Yeah. All right, so what do you see? We're getting close to winding up. So what do you see as the future for agriculture? What, can you see any bright lights in the next 30 years, or what's your crystal ball say? You are concerned about climate global
I'm
warming.
quite concerned about climate warming, yeah. It has potential big-term risks.
There is a trend that out in the wheat belt over the last 60 years.
I've talked to farmers, 90-year-old, said, what do you see? And they say, it's got drier and it's got hotter.
But
we've been here for long enough to know it.
But our yields have got better.
Yeah, but that's actually a consequence of drying because you're not getting waterlogging. And I had to explain this to a scientist from the eastern states. He said, why is it that people are growing more crops in the high rainfall zone? And I said, because there's less waterlogging. Oh, yeah.
But not only that, there's also you can plant in March.
That's right.
And you can dry the soil profile out.
Well, it's dry sowing, yeah. But the reality is, and you've only got to see it this year, happened again in the high rainfall zone. Anywhere that was waterlogged, bang, no crop, just gone.
Yeah.
So the less and less waterlogging you have, the better the returns are going to be. And we used to have, I can remember going out to Merredin back in the mid-80s, and there were paddocks that were waterlogged out in Merredin. Well, you don't see that anymore.
So, but another reason you don't see that is because the soil structure was damaged. You know, there's a beautiful video. In the US, there's a guy that's got no-till soils. Google it, and he holds up 2 jars. One jar's from a soil that's been ploughed for 60 years, and the other one is no-tilled for 60 years. He puts a little bit of soil in the top of one and the top of the other, and you see the one that's been ploughed, when it puts into water, it just completely explodes. Water bubbles go into it and it completely shatters, loses all the structure. And then and the other one just stays like it and just soaks the water in. And then he's got another tray. Let me finish. I know you're not paying. You can be a little bit of a go. And then he's got a tray where he's got the same thing and he puts the water on the top and the water runs off the one that's been ploughed. And the one that hasn't been ploughed, water soaks through it. So when you get this better water distribution, water soaking in, you've then got better plant soil water relations as opposed to, it can be muddy or it can be dry and won't run in. So soil structure is a big part of this story as well.
Well, there's a bit of that I'm sure goes on. But on some of the soils I can remember, these were ones where you basically had a hard pan at only 20 or 30 centimetres down. And unless you're actually breaking that up, you're not going to do much. And those same places still exist and they still get water now as what they did back then. So, yeah, I actually think that you've actually regulated this, or we're actually getting more evaporation in wintertime, which is taking away the excess water as well. So there's a combination of all these factors occurring. So, yes, it will happen. I don't think until the average global temperature for a 30-year period hits about 1.5 degrees, and so far it's only been for 2 years. When it's been 30 years, then we will see the effects, because at that point the oceans have warmed sufficiently to have it. So that's sort of a more long-term. I think the interesting thing that's going to happen is people are talking about drone technology. I don't think they've actually taken drone technology to the ultimate conclusion yet, and it's still very much focused on relatively small issues around drone. But if you watch what's happening in the Ukraine war and how Ukraine is using drones against Russia, and you sit there and say, okay, so how are they using their swarms? How are you using a drone mothership? How are they using it to AI to connect up all of the drones together? How are they? There's actually a technological revolution that's going to occur in drone technology within probably 30 years, well, probably 10 years actually, where you will see this. It won't be a swarm, but a very regulated process of selective management of crops using drones working in collaboration with each other with AI, and it doesn't require a single operator. And that will be another revolution. So that's part of the autonomous revolution that I think will happen. And you've already got those farms, research farms in the United States where autonomous technology is working completely, where you do not have to have a single person in the field.
I've got a friend.
And it's all pre-programmed in on a computer.
I've got a farmer friend that's doing that. He's got a tractor that just plants his crop on its own. He doesn't even need
Exactly. So, if you combine that with the fact that and I don't know that GPS technology through space will actually be the way to go. I think it'll be individual GPS technology on farm will be a far more reliable process in the future, because all the Starlink satellites are all going to burn up when they re-enter the atmosphere, and a lot of the GPS ones are going to be blasted out by China or whatever the case may be. Then we're gonna see back to individual GPS technology on farm.
I think we'll go back to the horse and the plough.
Oh, a few people might, when you can't buy fuel. So that might be one of the things. There are other risks that very few people are aware of, some of the big picture risks that are actually there. Do you realise that your rubber, your tyres on your car, all comes from two major sources, southern China and Southeast Asia?
Right.
Brazil used to be number one till they got a disease that wiped out 95% of their production.
Talk about synthetic rubber?
No, natural rubber. Right? Natural rubber. So natural rubber is produced in southern China and parts of Vietnam and Southeast Asia, right? That disease that wiped out the rubber plantations in Brazil, if it gets into Southeast Asia, you can write off pretty much all natural rubber production.
But there will be one plant that will survive it.
It might be, but they still haven't found it in Brazil.
Well, I was in Congo and rubber used to be a big thing there, Zaire. But
so that's actually one of the risks people are talking about now is biosecurity risks associated with things like critical crops in the system.
So also the pothole shot borer, you would be familiar with that. Yeah, but with technology, think we've got a pretty good chance of managing some of these things. Apparently a nematode, UWA guy was talking about a nematode that would help with that. All right, so we could talk to you forever, but our time's pretty much done. But thank you for that. Is there any sort of concluding comments that you'd like to make on agriculture in the future in Western Australia or globally?
Not really. I think it'll just basically do the transformation it's always done. I've always said that Western Australia largely is a technology taker, technology maker, and that's one of the biggest risks that it has because its genes come from elsewhere, its fertiliser comes from elsewhere, its pesticides come from elsewhere. The machinery still comes because we don't produce hardly any anymore. So our risk is the fact that being a technology taker is to make sure that the supply chains actually allow the ongoing development of the adaptation that occurs at this state level. And not many people have realised, you cut off fuel supplies, bang, it dies overnight.
Yeah.
You cut off nitrogen supplies, bang, it pretty much, a lot of broadacre systems go. You shut off the supply of genes, it takes a lot longer, but it slowly starts to degrade. So you've just got to be really switched on to the sort of critical issues associated with those things.
And energy drives nitrogen production as well. That's a tricky one. We're going to...
Well, it's going to transform. It's not that far away from transformation.
Can you see solar panels making it with excess energy that doesn't want to go into the grid because you'll get charged for putting it in the grid, being converted into ammonia?
Oh, yeah. Look, there was recently a patent released which shows how to make ammonia at room temperature and pressure, right? All you need is a supply of hydrogen. Now, that is a revolution because the old harbour process is very, very energy expensive. If you make these cheap enzymes which can actually do this, then basically nitrogen becomes like, cent a gram. It becomes really, really, really cheap. The biggest risk then is we have too much nitrogen in the world, and that feeds all sorts of dramatic effects.
Right. Well, I think that's another podcast, but we are agriculturalists, not but you do have a passion for chemistry, and you do love reading papers.
The reality is agriculture affects the environment, positive and negative.
You've got to remember that CO2 is sucked out by plants, and they love higher levels of CO2. So, as we know, there is a bit of rubberiness about models and about biology and life, and but you've certainly been at the pointy end of a lot of interesting issues, David, and really appreciate you taking the time to come and speak to us today. So thank you very much.
My pleasure.
Thanks for watching this episode, brought to you by 4Farmers.